Hello everyone, in this video you will learn something about a very cool enzyme. It's called urethanase and it can degrade plastic. I just watched a really funny video from Greenpeace. It's called "Whale Drew, Your DDs," because the whale is from New Zealand. And actually it's not so funny. The whale is dead because it ate too much plastic, and plastic pollution is a big problem in the ocean.
Thankfully, there are people who are doing something about it. Today, I'm meeting one of them, here in Greifswald at the third oldest university of Germany. His name is Professor Uwe Bornscheuer, and he is doing research on an enzyme that can degrade plastic, the urethanase.
You've got an enzyme in your lab that can degrade plastic. What's an enzyme?
Enzymes are the natural biocatalysts, and they are a subclass of proteins. All proteins are composed of the 20 natural amino acids, and the sequence of the amino acids determines at the end their function. And the string of these amino acids folds into a three-dimensional structure, and within the structure there's a so-called active site, and this is where the chemical reaction takes place and is catalyzed by the enzyme.
But plastic is not organic material, it's all artificial. So how can an enzyme do anything about it?
And that is one of the challenges, because natural polymers like cellulose are composed of sugars, and during natural evolution, enzymes in many microorganisms or organisms have evolved to degrade this complex biopolymer cellulose into the building block glucose, and then they can live on and eat the glucose. But all plastic polymers were invented by chemists around 100 years ago, and therefore natural evolution simply had no time to evolve enzymes to degrade these artificial polymers. And that is why it's so difficult to find enzymes which can recycle polymers.
Yeah, that makes sense, because otherwise I guess microbes would have already used these artificial polymers as a source of their food.
Yes, and there is one example where scientists from Japan identified a microorganism on the dump site of plastic waste, and it turns out that this microorganism can secrete two enzymes, and one enzyme can chew on the plastic, which is polyethylene terephthalate. We all know this from the plastic bottles, called PET, but this enzyme is extremely slow, and it can only degrade part of the plastic. But that is maybe the only example where there is a link between evolution of enzymes and degradation of plastics.
Wow, that's super fascinating.
Yes, but on the other hand, there are intrinsic issues with the different types of plastics. So there is of course polyethylene, polypropylene, polyvinyl chloride, polystyrene, polyesters and polyamides. We know this as nylon. And polyurethanes, we know this, for instance, the mattress you sleep on is composed of polyurethanes. And the challenge is that for each of the different types of chemical bonds in these molecules, you need different enzymes, and maybe a cocktail of enzymes to degrade them.
So you were talking about polycarbon chains, and you mentioned different bonds. You mentioned the ether bond, and then you mentioned ester, you mentioned urethane, and they're also occurring in nature, and they're like polypeptide chains and maybe polyfatty acid chains. And those chains are also polycarbon chains with some other atoms in between, and they're not a problem for the environment. So what exactly makes now the plastic, or some of the plastic, so difficult for nature to degrade?
Yeah, that is a very good question. If we look at a polyester, like the PET bottle, then we have clear chemical bonds which are known to enzymes. These are the so-called ester bonds. We also have this in olive oil, which is a triglyceride, so natural lipase, which we also have in our body, can hydrolyze this olive oil into fatty acids and glycerol, and then we gain energy from that. The problem is that even if an ester bond is in one of these chemical polymers like PET, the enzyme must have access to the ester bond, which is of course hidden in this long and very hydrophobic polymer. So it doesn't help to just have an enzyme which is an esterase and can hydrolyze esters. It must get access to the chemical bond and then can start hydrolysis. This is of course possible, and now enzymes have been engineered. That means scientists like me have improved the performance of the enzymes, and the PET bottle can be degraded with specific enzymes quite efficiently. The challenge is that, for instance, polyethylene, which is widely used as packaging material, is a hydrocarbon. It has no functional group where any enzyme can directly act on. And in addition, you will never get the monomers. So what we want to do with recycling is, if we take PET, we have an enzyme or a chemical process, we get terephthalic acid and ethylene glycol, and then the chemist can make a new polymer and don't have to use petrol sources again to make it. But for polyethylene, this is made from a gas called ethylene, and then all the double bonds disappear in the polymer, and then we have the issue, how can we find a microorganism or a set of enzymes which can act on this specific polymer.
Okay, now I got it. So if you want to recycle it, you need to break down these polycarbon chains into smaller units.
Yes.
And these units can then be recycled into something new.
Yeah, and that is also possible for nylons, polyamides, because they contain amide bonds. This is like in a protein where the amino acids are also connected with an amide bond. But scientists have found only a handful of so-called nylonases so far, and they are not very efficient yet. And the same is for the urethane, polyurethane material. This contains a so-called carbamate bond, and we have discovered 3 years ago the first enzymes able to act on these specific bonds in a so-called metagenomic library, which means you have several thousands of genes encoding unknown enzymes, and eventually one of these genes encodes an urethanase which was not described before, and we could find this, and now we try to improve these enzymes so that they can act on different polyurethanes.
Cool. Okay, you mentioned a library for DNA. Is that like a library with books where I can go and get a library card and then just rent DNA?
It is not as simple as that, but the analogy to a library full of books is okay, because imagine you want to find a book with a certain sentence in it, and then you're in front of our university library. They have hundreds of thousands of books, and you have to find this book with the right sentence. So this is extremely time consuming for you. You have to go through all books, read all of them, and eventually you can mark, I found it. And the metagenome library we analyzed comes from an industrial production site where polyurethane was produced over decades. And the idea behind that is that in the soil next to the factory there might be microorganisms which already contain enzymes which can act on the polyurethane bond. The problem is they have not been described before, so we had no idea how the protein itself looks like, how the active site might look like, and then we need a very good assay to find the desired enzyme in a library of several million clones. That means you take the metagenomic library, isolate all the DNA of the many unknown organisms, cuts this into smaller pieces, puts this into a microbial host, which is Escherichia coli, a bacterium, and then if you are lucky, one of these E. coli contain the piece of DNA which encodes the desired enzyme, and you find this with your assay, and this is like winning in the lottery, and this is of course major success, and at the same time there is always a chance that you find nothing.
That sounds like a lot of screening of E. coli and a lot of cloning. I guess you got a lot of students in your lab who do that for you.
Yeah, in fact that was a postdoc, but of course we also have very motivated students, and at the end of the day this sounds like an enormous amount of work, but it was done within several weeks, and that is still okay. And as long as you find something and have the breakthrough, then everybody is happy, even if this was a lot of work. It really becomes frustrating if you have a project where you spend 3 years and you still have no breakthrough.
Yeah, I can relate to that. During my PhD, I had a project like that. Great. Well, congratulations. This is great that you found this enzyme, but I'm still not fully sure. Plastic is so smooth on the surface, so how can this enzyme start to chew on the plastic polymers?
Yeah, that is also a challenge, and this is related to the material science, because the very hydrophobic surface cannot easily be accessible by the enzyme, because enzymes like hydrophilic environment, like water, like in our cells, which is aqueous. And then also plastics occur in crystalline and amorphous forms, and the enzymes can only chew on the amorphous form because that is more flexible. And that also explains why the recycling of PET with an enzyme must be performed at around 70 degrees centigrade,
Oof.
really high temperature for an enzyme, because only then the crystalline part becomes amorphous and can be degraded, so that we have a full recycling of the PET and not just 30% at room temperature.
That makes sense. Thank you. And now you found this urethanase degrader, this plastic degrader enzyme, and you probably found several. So how do you know which one is best? And if they're not good enough, how do you improve them?
Yeah, that is of course a major part of my research. So if you want to improve an enzyme, you must understand it better. So on one hand, we determine the crystal structure of the protein. So someone has to crystallize the protein, that is done by my colleague. And then you determine the three-dimensional structure, and then of course you can try to figure out which amino acids shall I change to another one to make it more stable or more active or both. And then of course there are many types of combinations of mutations which you have to introduce, so this you cannot do just manually by one PhD student. And in order to speed this up, we have a so-called high throughput screening platform, that is a robotic platform here. And this device can analyze up to 20,000 variants per week. So we have E. coli clones which contain the modified enzyme, and of course 20,000 different ones, and we don't know which is the best one. And then they are cultivated in this machine, the enzyme is produced, the enzyme is isolated, and then we can measure their activity in these machines and figure out, is this enzyme variant more thermostable, is this one more active, and eventually we can combine these features, and this is done by this unique automated robotic platform.
It's so cool. Look, the robot's got goggle eyes. So this is great, Uwe. You found this plastic degrader urethanase enzyme, and now you basically solved the problem. So there's a lot of microplastic pollution in the sea, and couldn't you just now go and put your enzyme into the sea?
That is possible. We can put the enzyme into the Baltic Sea, into the ocean, but it would be not a good idea. First of all, it will be diluted a lot in the big ocean, so we have to produce huge amounts of the enzymes, and when we put them into the ocean, then this is just a nitrogen and carbon source for all the microorganisms living in the ocean. So the chance that this enzyme ends up on a specific plastic material, microplastic or waste bag, is very little. The second point is, of course, as mentioned before, we have different types of plastics, so we need various different enzymes for all the different plastic materials, and for many of them we still don't know how they can be degraded with the help of enzymes. And a further problem is, as mentioned before, for PET, the temperature is too low to change the polymer structure into an amorphous structure. So we cannot heat up the ocean to 70 degrees, obviously, and even if the ocean would have 30 degrees like in tropical countries or so, this would not be enough, even with an efficient enzyme, to do this. And the alternative that we design microorganisms which are specifically constructed by scientists to degrade a certain plastic and throw them in the ocean because they can live and multiply there, this is a genetically modified organism, so this is not allowed. So the bad news is, I think there's very little we can do about plastic waste in the ocean, whether this is macroplastic or whether this is microplastic.
And you know, I'm just thinking if you would do that, that would probably be a problem for your boat, because you've got a sailboat and it's also made out of plastic.
Yeah, of course, I have a sailing boat, but I think there is no microbe on Earth yet which can degrade such a complex polymer, so I have good chances that I can keep my sailing boat for a few other years.
Because it's fiberglass, isn't it?
That's fiberglass, and that is extremely difficult to access, even by chemical means. For
I
example.
see. Well, then can go sailing.
Yeah.
Thank you so much, Uwe. I learned so much today, and I'm just wondering, so now avoiding plastic, probably we cannot avoid it altogether because we use it for clothes and housing and so on, but what is the message we should give to the listeners?
Yeah, the message is there are many reasons why we have to use plastic, and there are good reasons, and performance of these materials, or insulation and many other things, and also for our clothing. But when you use them, never discard them after use into the environment, and whenever possible use plastic which can be recycled, and of course don't use single-use plastics.
Don't use single-use plastic and don't litter.